Today's topic is meiosis. As usual, here's Google's gift to me, a nice picture showing the whole process from start to finish. And by the time we're done, this should all make sense to you. All right, here we go. So some general vocabulary. Somatic cells, these are body cells that are diploid in number. For example, blood cells, lung cells, muscle cells, heart, everything, almost everything, right? Gametes, those are your sex cells. These are haploid, for example, your egg and your sperm. Autosomes, these carry traits that make you who you are, and these are from chromosomes 1 to 44, or pairs 1 to 22. And then sex chromosomes are, again, a pair of chromosomes that carry traits that help you um, determine certain things about you, but also, most importantly, help to determine your gender. So if you are XX, you're a girl, and if you're XY, you are a boy. All right, some more vocab. A karyotype. This is a diagram that shows the number and visual appearance of your chromosomes, right? Diploid is a cell that has two full sets of chromosomes, right? So 2N. In human or diploid number is 46. Haploid is having just one full set, right? So when you look at this karyotype to the side, is this a diploid or is it a haploid? Tell me in class, right? So haploid is just having one full set. I guess I could have found a picture of that. All right, some more sexual reproduction. This is pretty much how humans get here, all right? Two parents get together to produce an offspring. And then fertilization is the actual fusion of egg and sperm to form a zygote. So let's get into meiosis. Meiosis is the process of cell division that makes gametes in the gonads. So this happens in the ovaries of girls and the testes of boys. It produces haploid sex cells, so eggs in girls and sperm in boys. Um, and these haploid cells have half the number of chromosomes. And this is a very important process because if the gametes were not halved, then we would be walking around with like two heads and four arms, and we'd be walking around with a double set of information. So this is a very important process, and it is necessary for sexual reproduction to occur. All right. Um, so in order to create cells with half the information, meiotic division happens twice. So we have meiosis 1 and meiosis 2. In meiosis 1, we have the separation of homologous chromosomes, right? Um, and then in meiosis 2, we have the separation of sister chromatids. And I believe I went over this vocab already. If I didn't, um, just let me know if that language is confusing you. So in meiosis, basically the you have one duplication and two divisions, so you end up with half the original number. So before meiosis one, we have interphase. Remember, just like mitosis, interphase is the growth phase of a cell cycle. This is when the cell is growing, it's making proteins, and it's doing other functions like normal functions. Interphase is divided into three parts, if you remember. So meiosis one pretty much is mitosis, right? The growth phase one, or gap phase, if that's what I used, is when cell grows and makes proteins, and most of the proteins are made in that stage. The synthesis phase, this is when DNA replication occurs, so the same DNA replication that we talked about earlier. And then growth 2, or G2, is when you have a little bit more growth and more protein synthesis. So at the end of interphase, the cell has two duplicated copies of every chromosome, right? So a cell that has, let's say 7 pairs is going to end up with 14 pairs, right? Because everything is going to be duplicated at the end of the interface, right? Um, so, so again, if you started with one of each, you're going to end up with two of each. If you started with two of each, you're going to end up with four of each. So duplication is going to happen, right? And that probably should say 4n. All right. So then we have prophase one. So after the DNA duplicates, right, we go to prophase one. Prophase one is where the nuclear membrane breaks down, centrioles separate and make spindle fibers, homologous chromosomes pair up and become visible, right? And they typically pair up in tetrads, a cluster of 4 chromatids. And at this point, crossing over may occur. Crossing over is when the chromosomes are lined up together and the chromosomes can pretty much cross over each other and get tangled. When this happens, they swap pieces of DNA, and this process creates new combinations of genes, right? So you ever notice like a kid could have all of mom's face, all of mom's face, and then whoops, that kid has dad's nose or something like that. So that is how that could happen. Um, then we move on to metaphase. Metaphase is when the chromosomes line up in the middle of the cell in pairs, right? So metaphase is pretty easy to remember because M and middle, meta. Then the next phase is anaphase. Homologous pairs separate, right? Um, then we've got telophase and cytokinesis. So telophase is when the the nucleus separates pretty much, and cytokinesis is when the cytoplasm separates. Then we move on to prophase 2. Please note we did not go back to interphase, which means the cells did not duplicate again. So we're picking it up at prophase 2, right? The spindle fibers form and attach to the centromeres of the sister chromatids. Then they line up in the middle, but now, as you can see, instead of being in tetrads, they're just in a single file in pairs, right? Then that pretty much it starts getting pulled to each end of the cell, anaphase. Then we've got telophase and cytokinesis. So the splitting of the nucleus and then the splitting of the cytoplasm. All right. So sometimes they're shown together, but remember that they are two different processes. If the cytoplasm is not completely separated, then cytokinesis did not occur. You just have one big fat cell with two nuclei if cytokinesis does not occur. And now this should make a lot more sense. So we start off with the cell doing its business. Then it goes through interphase where it makes a copy of everything. Then it goes through prophase 1, metaphase 1, anaphase 1, telophase 1, and then back into prophase 2, metaphase 2, anaphase 2, and cytokinesis. So that's meiosis in a nutshell. Notice the me in meiosis because that's what makes little me's. That's what makes babies. Big difference between the two is you start off with the same thing, what you end up with. So in mitosis, whatever you start off with is what you're going to end up with because you have a duplication followed by a division. But in meiosis, you have one duplication followed by two divisions, so you end up with half of what you started with. All right. So non-disjunction is something that could happen in meiosis. It could happen at any point in meiosis 1 or 2. Ultimately, it is when the chromosomes don't separate properly. And when they don't separate properly, they end up leading to a cell that pretty much has either too much or too little chromosomes. Proper Popular examples of non-disjunction are, for example, what happens with Down syndrome, the most common non-disjunction, and I think the only one that really leads to survival. You can have non-disjunction of chromosome 18, trisomy 18, but it usually leads to death sooner or later. So Down syndrome is when you have extra copy of chromosome 21, either a whole extra copy or just a little bit part off. And so that's why there is this spectrum of Down syndrome. You could have it mildly or you could have it a lot. It depends on how much of that extra chromosome that you got. And so this is a karyotype. And looking at this, I can tell you that this is a boy because at the X and Y, this is a Y and this is the X. And I can see that he has Down syndrome. Another type of non-disjunction is the non-disjunction of the X chromosome. Boys often suffer from what's called Klinefelter syndrome, where they have two X's and a Y. Other examples that involve the non-disjunction of X chromosome are Turner syndrome, and triple X syndrome. So Turner syndrome is when you have only one X and it is the only viable monosomy. So it's the only non-disjunction where you're going to have one copy of a cell and of a chromosome and still survive. And then triple X syndrome, which is kind of like the girl version of Klinefelter's, is when you have three X's as a girl. Want you guys to look carefully at this chart. I want you to redraw it in your notebook, and I want you to put these things where they should go. All right? None of this should really give you any trouble. I'm just gonna say pollen is plant sperm, and that should help you put that where it's supposed to go. All right. So make sure this is filled in. I will be checking for this in your notes. Um, so make sure it's filled in. All right. You could preview our next topic. See you in class.